Alright. So there's my parent DNA. When that replicates, each one of those strands is going to act as the template for the new one, right? So half of the molecule is going to be that original strand, and the other half is going to be the new strand. And the second round of division, same thing. Each one of these strands is going to end up in one of the new molecules, right? And then it's going to act as a template for the new DNA that gets added. So I know I'm not an artist, but something like that. Okay, so we talked about that Messelson and Stahl experiment yesterday, where we used the heavy versus the light nitrogen, right? We were able to figure out that that's the basic process by which DNA replicates, right? So do I feel pretty good with that before we move on to new shift today? Okay, so I want to get to a website that I just realized might use Okay. That's really sad because it was a really good tutorial. So we'll see if it works now. But before we get there, we'll take some notes anyways. Makes me so sad.
Why is Flash gone?
Yeah, it was definitely like a security issue, I guess.
I thought it was just because Adobe decided to stop.
I mean, they did.
They're switching to HTML. Enable Flash.
I mean, you can still enable it. It's just.
I don't know if you can on your Chromebook. Can you? Yeah, I'm pretty sure Chrome got rid of it. Well, we'll try it out. We'll worry about that when we get there. So, when we look at DNA replication, and this is really specifically in eukaryotes, so organisms that have multiple linear chromosomes like us, it's going to start in multiple locations because we just have so much DNA. If we want to copy it in a timely manner, we're going to have to start at multiple locations. Okay. We call those, and there's a really large team of enzymes that we're going to look at that coordinates this process. So one of the objectives today is to be able to list the enzymes in order that they function in DNA replication and matching them to their respective jobs. So there'll be a lot of enzymes to learn about today and tomorrow. So, a couple things about these starting points. Replication always starts at what we call the origin of replication. It's pretty straightforward, right? Origin means like the beginning of replication. So you'll notice these dots are there every so often along the chromosome. So there's like a specific sequence or set of sequences of DNA that allow the enzymes to attach and start the replication process. We call each one of these points of replication a replication bubble. And so I know these are small, but when this process starts, replication is going to go in both directions along the strand, right? So we're going to go this way, and we're going to go that way. what we're going to be focusing on in most of the diagrams here is 1/2 of that replication bubble. Again, this is kind of tiny, but we call that a replication fork. So 1/2 of the bubble is the fork. So when we look at most of the diagrams today, they're going to show just one side of the bubble. They're going to show a fork. And eventually those bubbles are going to run into each other, right? And when they do, we're then going to end up with our two daughter DNA molecules. That's half old DNA and half new DNA. So when we're done talking about replication, you should be able to name each one of these proteins that's shown up here, and you should be able to kind of tell us what their jobs are. So that's definitely something you want to kind of keep an eye out for and highlight in your notes as we go through the process. So first step of DNA is that we have to unwind and separate the strands. Right? We mentioned that yesterday in the very simplified version of replication we looked at. And that's where our very first enzyme comes into play. So helicase. In our diagram there, it's these yellow blobs. Right here and right here. So they're going to find those origins of replication, and they're going to start to separate and unwind our DNA strands. Okay. Notice they are going in both directions. So this is two replication forks, so 1, 2, on either side of our bubble. What are they breaking as they separate those strands? Hydrogen bonds, right? So once we separate them, we do not want them to go back together. So here's the second important set of proteins we're gonna name here, which are called single-stranded binding proteins. We usually just abbreviate that SSB protein.
So these two same binding proteins, do they like prevent the
Yeah. So you'll see them, they're these little light blue circles here. They're not shown on this side of the bubble for whatever reason, but these little blue things here are the binding proteins, and so they're going to keep those strands separated for us so that they don't just go right back together and form their hydrogen bonds again. So those are the first two main characters in the process. So after we separate the strands, then we're going to start adding our nucleotides. And when that happens, of course, we are adding complementary base pairs, right? So as we go along, if we have a G, we know that we need to add a C. And if we have an A, we know we need to add a T. And this is done by DNA polymerase. There's actually multiple DNA polymerases that we've discovered, and they're numbered in order of their discovery. So the version that does this, that adds most of the bases in DNA replication, is DNA polymerase 3. All right, I'm going to separate this name into 3 parts. Want you to talk in your group, work backwards. What does this part mean? What does this part mean? What does this part mean?
Got five there.
Yeah. That's like.
ACE tells us that it's an enzyme. Yeah, so remember most enzymes end in ACE. Polymer, what's a polymer? A chain of monomers. And then we're specifically dealing with DNA. Okay. So this is an enzyme that puts together polymers of DNA. So it's adding nucleotides together, right? It's actually making or building the DNA polymer. Okay, so that's why it's called DNA polymerase. Okay. I'm going to actually back up for a second. Let's talk about this name, helicase. ASE, again, tells us it's an enzyme. What does the first part of that name refer to? Helix. So this enzyme is breaking apart the helix. Okay, so all of these names do, like, they mean something, right? So that can kind of, you keep that in mind, that can help you remind you what their jobs are. Okay, so when I went from PowerPoint to Google Slides, it got kind of messed up. But this is supposed to be DNA polymerase 3, so it's adding my base pairs there. So again, C is going to pair with G, and T is going to pair with A, and G is going to pair with C. Okay. Notice, again, my DNA strands are very obviously anti-parallel here, right? Yeah. Yeah, I don't know what happened there. I lost it in my animations, I think. So anytime we're doing a synthesis reaction, because that's what we're doing here, right? We're building, what does it require? Well, energy at least, right? Requires energy. And actually, we're going to talk about ATP first. Where does the energy for bonding usually come from? ATP, when we do what to it? Specifically break what apart? The phosphate group. Okay. Hey, what does this look like?
Who said a circle?
A circle. It does look like a circle. What do we have here, though? A phosphate, a sugar, and a base. That's a nucleotide. A nucleotide.
I'm too tired.
It's a yeah, it's just a nucleotide with 2 extra phosphates on it, right? That's normally how we get energy, is breaking off one phosphate group. What ends up happening with replication is we end up breaking off 2 phosphate groups, and we just incorporate it right into the DNA molecule. Okay? Pretty cool. So instead of just breaking off the first one, we break off both of the phosphate groups, and that directly puts it into the DNA strand then. Okay. So when those nucleotides come in to pair with the template strand, they actually come in as nucleosides. So they come in with those 3 phosphate groups. And when DNA polymerase adds them to the molecule, it's going to break off those last 2 phosphates. And that's where the energy is coming from to bind those nucleotides together. And we're going to look a little bit more at that on the next slide. But that's the general idea. So nucleotides come with their own energy source attached to them already, those phosphate groups. Okay. Are there other energy nucleotides? Yes. So we don't just have ATP, we also have GTP and So there's that breaking off to make our key nucleotide. TTP. Okay.
Yeah.
And CTP. Okay. Yeah.
Where did the protein get?
I mean, add into the cytoplasm. They'll eventually, like, this will get regenerated, right? So when we make our next nucleoside, we'll add the phosphate groups back on. So there's other enzymes that will put them back on. They just get recycled. We do a lot of recycling inside the cell. Okay. So a little bit of text to go along with that. So when nucleotides are arriving at the scene of replication, they're actually coming in as nucleosides. So that's just a nucleotide with the extra phosphates on it, right? So we have our DNA nucleotides and we have 2 extra phosphates attached to it. So we know that there's enzymes and processes that make ATP. There's also enzymes and processes that will make these other types. Actually, the Krebs cycle usually makes GTP. And so that's important because those phosphate groups are going to provide the energy for bonding. And so I don't know if you need, like, I feel like this is repetitive. The DNA bases are arriving with their own energy source. Okay. And the enzyme that's going to add them is going to be DNA polymerase 3. Right? So we already mentioned. So you feel pretty good with that? Any questions on that? Okay. This also directly affects the direction or the end of the molecule that we can add these nucleotides to. Okay. So we are adding, when we break these apart, we're adding the phosphate group to the growing DNA strand. So what end of the strand are we going to add this phosphate to? 5' or the 3'? The 3'. We have to add the phosphate to the sugar, right? So whenever we're building a DNA molecule, we always have to add these nucleotides onto the 3' end. Okay. So, like we just mentioned, whenever we add bases, we can only add new nucleotides to the 3' end of a growing strand because we have to attach it by the phosphate group. So in this case, I couldn't add nucleotide. I know this is off the screen up here. I couldn't add a nucleotide here because I have to attach it by the phosphate. So the phosphate has to add on to the 3' end, the free sugar end of the strand. It's also really important that DNA polymerase 3 cannot, it could not come here and add a nucleotide. It can only add a nucleotide if there's a starter there. Okay, so it would have to add the nucleotide here first, and then here, and then here, and then here. It couldn't just start anywhere randomly on that open strand of nucleotides. Okay, so it has to have a nucleotide to attach the phosphate to, right? You can't just attach it to nothing. And so this is red underlined here. Our strand is only going to grow from the 5' to 3' direction. Okay. So here's my original DNA molecule, 5' to 3'. This is building from the 5' to 3' direction. Okay, so it's only that's what we mean. It's only growing from the 5' to the 3' direction. We can only add those phosphates to the 3' end. The opposite way to say that is that we're reading it from the 3 to 5'. So we're building from 5 to 3. We're reading from 3 to 5. Okay, so we have that anti-throw going on there. Okay. I feel like we need a little bit of a break, so take a couple seconds to close your notes and just talk in your group about everything important that you remember so far.
We didn't do that.
I'm not. No.
You like speaking in front of people. Okay.
Okay. So, this is just a little animation showing this. So here's the new base that gets paired up. There's DNA polymerase. It's going to break off the extra phosphates and make that bond between the phosphate and the sugar, right? And it's going to move on to the next one. It's going to make sure it pairs properly. It's going to break the phosphate groups off and bond it to the sugar. So, we're building again, always on to the free 3' end, going from 5' to 3'. So, we're going to take a little look here at our replication bubble and therefore our replication fork. So, because DNA can only build from the 5' to 3' direction, we have this kind of interesting situation where as helicase unzips that DNA, one side can keep building and building and building and building and building and building because that 3' end is always going to be open to build on. Okay? We call that the leading strand. On the opposite side, though, we have to keep starting over as more and more of the strand becomes exposed. So, for example, I start here. I have to go from the 5' to the 3' direction. So I'm copying it this way. As it unzips, I'm going to have to start again and then go back in that direction. And then as more DNA becomes available, I have to start again. Okay. So one side gets copied continuously. The other side is going to get copied in these sections. So I end up, again, what we call the leading strand, which can keep going, and the lagging strand, which has to reset every time a new section of DNA is exposed. Okay. What do you notice as I go on either side of this replication bubble? What happens to the leading strand and the lagging strand side? They're getting flipped, right? So when helicase is going this way, the leading strand is going this way, the lagging strand is copying in parts. As we go this way through the bubble, the leading strand is down here, going continuously, and the lagging strand is getting copied in parts. Okay. So, anytime we start replicating DNA, there's another enzyme that has to act first. Because again, DNA polymerase can't start adding nucleotides until it has a 3' sugar end to add to. So before DNA polymerase can start adding and laying down those nucleotides, another enzyme here comes in called primase. So again, ACE means enzyme. What does it mean to prime something? To get it started, right, or prepare it, yeah. So primase has to lay down some nucleotides first so that DNA polymerase can take over and start adding on to the string, okay? What's a little bit interesting about primase is it actually doesn't lay down DNA nucleotides, it lays down RNA nucleotides. So these starting points here are actually RNA nucleotides, and then DNA polymerase takes over. So on my leading strand, I only have to do that once, and then DNA polymerase can keep on adding on to that 3' end as more and more DNA becomes available. If I'm on the lagging strand, I have to start over multiple times. So when I start copying here, primase lays down some RNA, and then DNA polymerase takes over. As more DNA opens up, primase has to start again, more DNA gets added. Primase has to start again, more DNA has to get added. So I end up with these fragments here. You know what? Sorry, I need to move some things around because that slide. I was thinking we were going to get to do that activity, and I'm not sure we are. So I won't go through it. So because we have all those segments there, we have to go back and fix that eventually. And so there's 2 enzymes that are going to be responsible for that. So the first one is going to be DNA polymerase 1. So DNA polymerase is also able to make a DNA polymer, but it's an editor protein. So you can think of DNA polymerase 3 as the builder, and DNA polymerase 1 as the editor. So it's going to be able to come through and be like, ooh, RNA, you shouldn't be there. I'm going to replace you with DNA. And so it's going to have to do that at every part of every one of those fragments that we made. And then the last enzyme here that's important is called ligase. You know what the word ligand means? A ligand is like a tie or a binder. And so ligase, in this case, is going to glue, after DNA polymerase goes back through and replaces the RNA with DNA, it's going to glue it together. So DNA polymerase 1, their editor, it's going to go back and replace the RNA with DNA. Ligase is going to join those fragments together so they're one complete sugar phosphate backbone. All right, so let's take a note break for a second. And in your groups, see if you can list all the enzymes so far that we talked about.
That's all. It's
They don't.
Yeah, no, right, like pasta. Right now, I let it down.
All right, let's see if we can kind of list them in order. So first enzyme that has to work is helicase because it breaks the hydrogen bonds, right? Separates the strands. Then the next one's not really an enzyme, but what's the next important protein that we talked about? Yeah, SSB, the stranded binding proteins, because those help to keep the strands apart from going back together. Then who would have to act next? Primates. Yeah, so this isn't the order we talked about, but primates would have to happen next because they have to lay down that RNA primer so that our next enzyme, DNA polymerase 3, can then join and start adding the DNA nucleotides, right? Make the actual new strand. When we are doing that, yeah, so we have the leading strand, which can just build continuously. We have that lagging strand, which is working in the opposite direction, has to do in parts. So what we need to do next is remove and replace those RNA primers. So who does that? DNA polymerase 1. And then lastly, we have to join those fragments together, which is done by ligase. One last term here that's important is that on the lagging strand, these sections are called Okazaki fragments. So you'll see that term for the lagging strand segments. Yeah, question?
Are we going to talk about polymerase 2 at some point?
So DNA polymerase. So we did most of this research like in E. coli actually. So the genetic mechanisms for that are really similar. And so they have a DNA polymerase 2. We actually don't have DNA polymerase 2. So it has a role in bacteria that we don't use it for in our own replication. So yeah.
What does it do?
I'm not actually sure. I'd have to look it up. I'm guessing it builds DNA. So yeah, we don't have DNA polymerase 2, but other organisms do. So we don't talk about it generally. Okay. All right. One more concept we need to get to today. I need to do that slide. Is what happens when we get to the end of the chromosome. Okay, so remember, one end of the chromosome is going to be the original 3' end. And so when we're building our DNA molecule, this is going to be an RNA primer. And since there's no nucleotides to the right of this, we can't replace it. We can't fix it. Okay. So what happens over time is the ends of the chromosome slowly can wear down. Okay, and so we think this might have to do partially with aging. There's been some studies that people that are under a lot of stress tend to have like premature shortening. So if you take chromosomes, so really the study was done comparing like African American populations with like white Americans. So especially in our society, usually African Americans are under a lot more stress on a daily basis. And so they actually when they look at these telomeres, on average, their telomeres are much shorter. So there's a relationship between stress and disease and aging with the shortening of the ends of the chromosomes. The ends of chromosomes are called telomeres. Okay, so if you know that term. So the telomeres get shorter and shorter and shorter every time the DNA replicates. So every time we have to divide and make new cells, then the chromosomes are going to get shorter and shorter and shorter. There is an enzyme that can help to re-lengthen the end of the chromosome. It tends to be active like in embryonic cells and in some stem cells that have to divide really often. They'll have this enzyme activated, and that enzyme is called telomerase. And so my lecture's all kind of messed up because I changed plans last minute. But those ends, again, are called telomeres. Generally there's like a protective cap here, but again, as we go through divisions and divisions and divisions, that gets worn away. In stem cells, and apparently a lot of cancer cells, this telomerase enzyme gets turned on, and it tends to extend telomere length. So it kind of loops. What happens is it loops the DNA over so that DNA polymerase can bind and recopy it. And so it does this looping so that there's a nucleotide to build off of. And so this repeating sequence here, for example, you can see you have this TTAA. If you looped that over, so it was anti-parallel, you would have TTAA, right, complementary. And so that gives them the DNA polymerase and nucleotide to add on to so that it can complete the copying correctly. And so that's going to add length back onto the end of the chromosome, the end of the telomere. So, and again, those are really high in stem cells, which are the cells that regenerate cells in your body, like your bone marrow. And they tend to be reactivated in cancer cells because cancer cells also divide really rapidly. So that's the notes I have for you for today. I'm going to assign you the quick check for tonight. Yeah, I wasn't going to originally, but well, I need to.
You don't need to do it.
No, I'm actually really busy.
Well, I don't think it'll take too much. You have like 5 minutes now. You could probably get started on a couple questions.
All right, well, let's do it. Exactly. What is it? No, it does not. Oh, is it just pop? DNA replication.
Yeah, so look for the 9.3 DNA quick check.
I really know.
I'm pretty sure that replication simulation runs on Flash.
Yeah, so that's your. We just discussed it.
Oh, that. What do you mean? I know, but like, Good point.
So
You forgive me for
Yes. Yes.
all he doesn't forgive me, he's happy. He just wants like answers.
What was that?
What is the origin?
Wait. Why was it? Wait, David,
I need to know that.
well, tell David he's wrong. Only question one all of the basic.
I would work.
Somebody got.
Oh.
Because the guy was like, it says that. It was like 5'9".
Arnie.
It was 11. Right? I don't know how feet work. Yeah, so it was like 11'9" or 11'10. 11 inches.